All right, just a quick review first. Here's our cell membrane picture. We know this is the fluid mosaic model of the cell membrane. It's mostly made out of what? Phospholipid bilayer with the blue things embedded in there. And what are all those blue things? Proteins. They're the mosaic part. They give it certain functions. Like this guy right here. What did we call this? Channel protein. A channel mostly for what? Two kinds of things. Yeah. Charged, polar kinds of things, and large molecules that have a hard time getting in otherwise. What do we call these things sticking out the top? Yeah, what was a more sciencey name for them though? Glycoproteins. Name tags. We might call them marker proteins. There's all kinds of names for them. But they identify your cells to what? To your immune system. So hey, don't mess with me. I'm one of the good guys. And then this down here we might have called a receptor. A receptor mostly for what? Hormones and other kinds of cell signals. There's all kinds of chemicals that your cells use to signal each other. So right now I want to say hormones. When we get to nerves and nerve cells, we're talking about neurotransmitters. And there's other kinds of signals we'll get to as we go through this class. One thing that we didn't talk much about that you might want to add into your notes, especially in animal cells, there's another chemical that's very important in your cell membrane, and it's cholesterol. Cholesterol helps to keep the membrane the right consistency. It is a fluid mosaic model, which tells you it should be kind of wet. Fluid, so it should be kind of liquidy, right? It should be able to change shape and move, and it does. Cholesterol helps to regulate that consistency of the membrane so that it's the way it needs to be. All right, let's move on. Let's talk some organelles today. Let's talk a little bit about the nucleus first. Did we already get to the nucleus in this class? Did we go through that pretty much? All right, so we'll hit this pretty fast in review. The nucleus is oftentimes considered an organelle. We know that's where the DNA is at. We know that's where the chromosomes are. We know DNA's only job, really, is to do what? Control everything by controlling what? It's the code for what? Proteins. DNA says, make these proteins, here's the proteins you need, and here's how to make them. And that really controls pretty much everything about your body. We know our membrane has our DNA surrounded by a double layer of phospholipids, a double membrane. So it's really 4 layers of phospholipids, right? It's a double bilayer. It's got pores in it because what things need to be able to get in and out of the nucleus pretty easily? Say again? RNA. What else? Ribosomes actually are made inside the nucleus. They have to get out. Where are most of the ribosomes going to be found at? Rough ER. But there's some in other places too, but most of them are rough ER. We know that dark spot in the nucleus is the nucleolus, and that's where the ribosomes are made. Our nucleus tends to be filled with something very similar to cytoplasm, nucleoplasm. And we talked about chromosomes. Realize that a chromosome is a single molecule of DNA. One long molecule. Most of the time, are chromosomes visible in your cells? Nope. When's the only time they should be visible? Just before, during, and just after what? Cell division, mitosis, right? Grab this. Hello? Yes, ma'am. Okay. Will do. Okay. So, the purpose of a chromosome is really to do what? Why bother ever having chromosomes? What does it let you do easier? Not so much copy it, but after you copy, what does a chromosome let you do? It would be really hard to do without a chromosome. Separate it into equal, exact sets. when the DNA is in the form of a chromosome, is that DNA really working very well? Because it's coiled up, right? The enzymes that let things like replication and transcription happen, they can't get to it when it's all coiled up. Chromosomes are all about separating, making sure that each cell gets exact copies of all the DNA. Think of it like a spool of thread. That's what a chromosome really is. What's the actual spool? What does the DNA wrap around to make a chromosome? A chromosome is made of DNA and what? What's the answer to just about every question I ask you? Proteins. In eukaryotes specifically, proteins called histones, sort of like spools for the DNA to wrap around. Most of the time, your DNA is not in that form. Most of the time, it's in the form of this stuff right here, chromatin. That's unwound DNA. DNA that's working. Caitlin, they just called from the attendance office. They need you to come back. Don't know why. Sure. So chromatin is DNA in its working functional form, not in the form of a chromosome. Unwound DNA. You're fine. But we're talking about a chromosome. Most people think of an X. But really, what is that X? Two copies of the same chromosome, right? Because the DNA has already been copied by the time the chromosomes wind up. What's the more scientific name for that X-shaped structure? What kind of chromosome? Word you need to know. They're called bivalent, which is letting you know that's really two. Two copies. During mitosis, what's going to happen to that X? It's going to split into two pieces, right? Two V-shaped pieces. When these pieces are connected together, we call those pieces chromatids. Sometimes what kind of chromatids? What's the word we have in front of that word chromatid sometimes? Sister. They're identical copies of each other. Connected together in here at the middle, at this point called what? Centromere. We know that's going to split eventually. Moving on down a little bit. In us, humans, how many chromosomes do we typically have per cell? How many? 46. Two sets of what? 23. Well, you ladies have two sets of 23. What do we call it when you have two sets of a particular kind of chromosome? Diploid. That's a good thing. Us guys, for 22 of our sets, we're diploid. We have two pieces. But for the 23rd set, and by the way, what is that 23rd set of chromosomes called? What makes us different from you? What kind of chromosomes? Maybe. Us guys over here, you guys are segregated, by the way. Did you notice that? Alyssa's sort of the eyeball over here. Right? Segregated. What makes these guys over here different from you ladies over there? The sex chromosomes, right? X and Y, X and X. Over there. That makes you different. For us guys, that one set of chromosomes, that X and Y, they're not the same. If we get a messed up X chromosome, a mutated X, what happens? To us as guys, we have whatever that mutation is, right? Because there's no backup plan. You ladies have a backup plan, right? You've got an X from mom and an X from dad, and oftentimes you don't get genetic diseases that us guys will because you have a backup plan. Being diploid is a good thing. Protects you against mutations. That's important to think about. All right, one other thing I'll mention, in prokaryotes, what's the normal number of chromosomes? Okay. One. And Matt, what shape is it? Try again. What's the opposite of linear in terms of the chromosome? Somebody help them out. Circular, like loops. Ours are said to be linear. They're not straight lines, but they're not loose. All right, there we can see our nucleolus in the middle. That's where ribosomes get made. Our double nuclear membrane with pores for RNA and such to come out of. And notice how close it is up to the ER, the rough ER. Makes sense. It's another one of those examples of structure determines function. That ER is right where it needs to be for this to function properly. All right, let's get into some other organelles, what we call membrane-bound organelles. Vesicles, I'm going to use interchangeably with what other word? Vacuoles. They're basically storage containers. In a plant, the biggest part of the plant cell, other than maybe the wall, is what? It's a big vacuole in the middle. And mostly that's for storing what in a plant cell? Okay. Water. Can also store starch and other things, but big water vacuole. Realize, though, that not just plants have vacuoles. All kinds of cells have vacuoles. They're important for storage. They're also important for transport of things, moving things around the cell. And we're about to get to how that works. The ER, we've talked about over and over again. Endoplasmic reticulum. It's a folded, stacked set of membranes, tubular-shaped membranes. Can be attached to the nucleus. The ER we most of the time talk about is the rough ER, because it's covered with ribosomes. If you don't already know it, know that ribosomes make proteins. Those ribosomes are on top of the ER. It's another example of structure, function kind of stuff. In a factory, like an assembly line kind of factory, how do things move around inside that factory most of the time? On a conveyor belt. I want you to think of the ER like a conveyor belt. I want you to think of the ribosomes that make whatever the factory makes, and then the conveyor belt moves it around. After it's made, it needs to be maybe modified, maybe improved a little bit, and then eventually it needs to be what? It happens at the end of the assembly line. You box it up and you ship it out, right? We're about to get to how all those pieces work. But I want you to think of the cell sort of like a factory, an assembly line kind of factory. The rough ER, again, covered with ribosomes. It helps to ship cell products through the cell. It also helps to modify those cell products. When we say cell product up there, really what are we saying? Proteins. That's what cells make. Cells make proteins. There's lots and lots of rough ER in the pancreas and in the salivary glands. Now, let me make a point here. Does every cell have every organelle? Pretty much. But does every cell have the same amount of every organelle? No. Again, that's that whole structure function thing. If you're a cell that's making a lot of stuff to ship out, then you better have a lot of what two things? If you're making a lot of stuff to send to other cells, you better have a lot of rough ER and a lot of ribosomes. Those are the structures to do it. The pancreas, maybe the most underrated organ in your body, maybe the liver too. People don't know much about the liver. The pancreas makes almost every digestive enzyme that you have. But the food never makes it to the pancreas. Therefore, the pancreas, after it makes those enzymes, it has to ship them out to where? Where do the enzymes work at? In the small intestine. So that's why the pancreas is going to have lots of ribosomes, it's going to have lots of rough ER. Because it's making lots of proteins, and it's shipping them to somewhere else so that they'll work. The salivary glands, we just talked about this a minute ago when we were talking about the mucosal. What do they make a lot of that they ship out into your mouth? Which one? Which enzyme do we talk about found in saliva? Breaks down starch. Somebody help me out here. Who was the one person asking me about that one? See? Amylase. Breaks down starch found in your saliva. There's a lot of other places that are really loaded with the rough ER, but those are two good examples. The smooth ER doesn't have ribosomes. It's involved in a lot of stuff too, though. It makes steroids. You guys name two steroids for me. Testosterone, estrogen, cholesterol also is a steroid. They're mostly hormones. They have other functions in your body. So that being the case, where should there be a lot of smooth ER? Where's estrogen made, ladies? The ovaries. They should have cells of the ovaries should have a whole lot of smooth ER. Cells of the testes, other places in your body as well. They destroy toxins. What organ in your body helps you get rid of poisons? The liver. There's lots of smooth ER in there. There's a study that I heard about on the radio a couple of days ago that the livers of alcoholics have way more smooth ER than the livers of normal people. Because alcohol is what? It's poison. And it's your liver's job to get it out, to help you get rid of some of it. Also helps to regulate, this should say calcium ion, 2 words, concentrations. When we talk about muscles, we'll talk about how that works. It's mostly a muscle thing. Questions here? Let's skip that for a second. This slide, really important. This slide, we're looking at something that's big in your Moodle assignment. I guess it was in Moodle assignment 3. The what system? Endomembrane system. Whenever you see that root endo, means what? Inside. Membranes inside the cell. The endomembrane system I'm about to describe to you right now. Here's a ribosome. It's not a membrane. It's like a solid thing. Think of it like a tool. It's making a protein. If this is a protein, what's each one of those little balls? It's amino acids. So it makes a protein. The protein falls down into this ER. It's like a tunnel. One thing you ought to notice, when it goes down in there, what happens to it? What changed between here and here? Its shape. That's one thing that happens inside the ER, is that protein gets bent and twisted into the right shape so that it will what? Function, structure, function thing. That's one of the things that happens in the ER. It takes on the right shape. It may get some things added to it, like sugars or lipids and other things. So it's not just about moving it, it's about improving it, getting it ready to work. That's what happens in the ER. Eventually, though, that protein gets to the end of the ER, and this membrane starts to pinch. And remember, we call that pinching inward imagination. Eventually, this piece is going to touch this piece. And when phospholipid touches phospholipid, what happens? It's like welding them together, right? Joints. So the sides close. And now we just bagged it up. So we had the factory, the assembly line, and we made it to the end. Our cell product, our protein in this case, got bagged, and now it's ready to what? Ship out. This is the starting point of the endomembrane system. Before that bag goes anywhere else, most of the time, what other organelle does it go to next? Usually goes from ER, well, let's start at the top. Ribosome to ER, to vesicle or vacuole, to where? What comes next? The Golgi apparatus. And the Golgi apparatus is, again, it's a stack of bags, of vacuoles, membranous sacs. Most of those sacs come from the rough ER. In the Golgi apparatus, or Golgi complex, or Golgi body, they're all the same thing, whatever you want to call it. The proteins get modified, they may get improved, they may get sugars, lipids, different things added to them. But the main thing that I want you to think of when you hear Golgi apparatus is this word right here, exocytosis, which means what? Out of the cell. The Golgi body or Golgi apparatus sends things out of the cell to another cell. Sometimes we call that secretion. Its job is to move things out of the cell. This is basically one of the last parts of what we've been calling the endomembrane system. And I want to show you how that works. Here's Golgi apparatus. Remember, this is made of lots of those vesicles stacked up. And what's inside each one of those vesicles? A protein. A protein that the cell made that it needs to send somewhere else. So eventually, after that protein has been modified and it's ready to go, it breaks back off again, transport vesicle does, and it bumps into the cell membrane. What's that vesicle made out of in the first place? Phospholipids. What happens when phospholipid bumps into phospholipid? Confused. But what happened to that protein that was in the bag? It got pushed out. That's exocytosis. What just happened to the cell membrane? It got bigger. It grew. This is how the cell membrane grows. It grows when these vesicles from the rough ER and the Golgi apparatus collide with it and ship out some kind of cell product, some kind of protein. So the endomembrane system, this is pretty much it. We start with a ribosome, which is not a membranous organelle, but it makes proteins. Those go down into the rough ER, where the proteins get their shape changed. They may get modified slightly, and they get shipped. Eventually, the rough ER will form a vesicle around that protein, a secretory vesicle. Those will break off, stack up at the Golgi apparatus. And again, while the protein's there, it's getting modified, it's getting improved. Once again, a vesicle will break off of the protein inside, and that will lead to exocytosis. Exit from the cell. Push the protein out of the cell. That's what we call the endomembrane system. And at least in eukaryotes, that's what kind of property? That's a conserved property. It's found in which eukaryotes? All of them. Now, it's not found in prokaryotes because they don't have a lot of those structures, right? They don't have rough ERs and smooth ERs and all that kind of stuff. They don't have organelles. But at least in the eukaryotes, this same basic process works almost exactly the same way in all eukaryotic cells. It shows that eukaryotes are related to evolution. Questions? All right, the last couple of organelles I want to mention. Lysosomes are an important one. I think I might have mentioned this in here before. Anytime you see LYS or LYT together in this class, what should you think about? What does it mean? LYS, LYT. It's the root you're going to see a lot. It means to split, to cut, to break, to tear. So, lysosomes are about breaking down of things. And they can break down a lot of things. Sometimes it's old cells, worn out cells that your body needs to get rid of. Sometimes, for example, inside a white blood cell, white blood cell needs to break down. A virus, a bacteria, some kind of pathogen. So that's why we say destruction of pathogens. Say a tadpole, for example. How many of you caught tadpoles and watched them turn into frogs as kids? Put up your hand. You caught tadpoles and you watched them turn into frogs. 3 people out of the whole class. Wow. The tail of that tadpole, what happened to it? How did it go away? Fall off? The tail fall off? Just kind of got smaller and smaller, right? What was happening to the tail? These lysosomes are breaking it down. What's the tail mostly made out of? Take away the water, what's mostly in there? If we broke down Alex's arm here, what's mostly in there? What's the answer to almost all my questions, guys? Proteins. So if we broke down the tail, what do we have a bunch of after we break it down? If we're a tadpole. What do you get when you break down proteins? What does the frog turn and use those amino acids to do? Grow what? As the tail's going away, what's starting to show up? Leads. It's recycling. So sometimes you'll hear lysosomes called the recycling centers of your body. They may break down something in one place, but the pieces of the broken down stuff may get reused to build something else. So sometimes they're called the recycling centers. Occasionally your lysosomes can break down things you don't want them to. There's some research that shows that Alzheimer's disease may have something to do with lysosomes out of control in your brain. You might have seen a picture of somebody's brain with Alzheimer's. It's got big holes in it. There's pieces that are missing. They're gone. There's also pieces that are covered with this plaque-like materials, coating. The parts that are gone, the brain of a person with Alzheimer's weighs way less than a normal brain. Some of it's been broken down, and some people think it's a lysosome issue that may be causing it. The answer is they really don't know for sure, but it probably involves lysosomes in one way or another. What you need to know about lysosomes: they contain powerful enzymes to break things down. Hydrolytic. That does not mean they break down what? Doesn't mean they break down water. It means they use water to help them break things down. Water's a reactant. And again, the kinds of things they break down are maybe germs, pathogens, old, dead, worn out cells. They're involved in metamorphosis. Recycling would be a good word to put into your notes. They help to recycle things. They may break down one piece, but the parts get reused. Your body is very conservative in terms of the way it works. It's not going to waste things. It's going to use what it has and use it as efficiently as possible. All right, vacuoles and vesicles. We talked about those already. Just briefly mention them again. We're going to use those two words interchangeably. They store stuff. They're made of phospholipids. They may store water, like in a plant. They may store food. They may store toxins. They may store wastes. They can store all kinds of stuff. This word, when we get into our next unit on cell transport, is going to be an important word: turgor pressure. If you put a plant into fresh water, if you soak its roots in fresh water, what's going to happen with the water? Put a plant in fresh water. What happens? The water goes into the plant. What process moves the water into the plant, at least partially? What's the process that moves water mostly in nature? So again, osmosis. Osmosis moves water from where there's a higher concentration of water to where there's a lower concentration of water. But oftentimes we talk about it in terms of what's in the water. And this is really what X move assignment is about. Oftentimes we say osmosis moves water from where there's less solutes to where there's more solutes. What's the solute? That's something that's dissolved, like salt, sugar, anything that'll dissolve in the water. Well, if you put a plant in pure water, how many solutes are in the water? If it's pure water, not many, right? What's inside the plant? What kind of solutes are in the plant cells? Sugars, salts, all kinds of stuff. So the water moves in. And as the water moves in, what happens to those vacuoles in the center of each of those plant cells? As more and more water goes in, what happens to those vacuoles? They swell, right? Now, if this was your cells, if we put one of Matt's cells in pure water, what's going to happen to it? It's going to swell, swell, swell until eventually it's like a water balloon that you overfill. It's just going to pop, right? But what does a plant cell have that Matt's cells don't to keep that from happening? A wall. Eventually, what's going to build up in that cell so much that no more water can come in? Pressure. There's going to be a certain amount of pressure that's going to push the water back out, even though it's still want to come in. What's that going to make the plant cell do? Just think of it this way. A wall for a plant cell is sort of like what part of your body? It's like a skeleton. Because if those water vacuoles are full, they're pushing out on that wall, right? And it makes it really wet. Yeah, so the plant stands up. Skeleton. But if it's a really hot, dry day, what happens to the water in those vacuoles? It starts to go away, the pressure drops, and what happens to the wall? It's kind of saggy. Flaccid is a word for that. And the plant starts to wilt over. So the vacuoles are really important in plants for helping it to even just stand up. We're talking about plants, of course, that don't have wood here. All right, we'll talk about the mitochondria. This is one seen over and over again in this class. We know it's sometimes called a powerhouse. You've probably all heard that from elementary school. It makes ATP by doing cell respiration, aerobic cell respiration. I want you to know that word, aerobic. Meaning that it's got to have what? Oxygen. We need oxygen so that we can make ATP. Now that whole structure function thing comes into play again with the mitochondria. I'm going to go to a picture. I'll come back to this slide in a second. I want us to look at a picture. Here's what a mitochondria looks like. It's got two membranes. One on the outside that's nice and smooth. This one out here. But notice the one on the inside is all twisted, convoluted. What have we said in here before? When we see something that's all twisted up like that, what should we be thinking about? Surface area to volume ratio. Surface area. It increases the amount of surface area that you can fit in a certain size space. Turns out that inner membrane is where an important part of respiration happens, and you need a lot of space for that to happen. Where else in biology do we see that same kind of pattern? Twisted up stuff. Where else in your body do you see that pattern of something that's really, really twisted and convoluted? Your small intestines. Both the outside part, they're all twisted. But also if you were to look at the inner wall, they're going to look like that, right? It's all about increasing surface area. Where else? Where else in your body do you see all those twists and bends? The brain, right? The surface of the brain. To have the brain power you have right now, if it wasn't all twisted like that, your head would have to be huge. Most of us don't have that except for Julie, right? Most of us have no. It's just all the power in there. Regardless, why two membranes? Why bother with this outer one? Where do scientists think that, or at least originally, the outer one might have come from? What's the idea about where mitochondria came from? Yeah, it was basically a separate, what kind of cell? Prokaryotic cell, is the idea. And how did it get into a bigger one? Was eaten. What's the word for that? Or sciencey word there? What word actually means cell eating? Speak up, Colin. Phagocytosis. And when a cell eats something, when it engulfs it, something big and solid especially, what does it typically put around the thing that it ate? A bag, a membrane. So that's one idea about where that outer membrane came from, is that that inner membrane might have been the original bacterial membrane, and the outer one resulted from phagocytosis. Regardless, in this membrane, two main steps in aerobic respiration happen in this mitochondria. We'll talk about respiration here in a few weeks. The first step is called glycolysis. That does not happen in the mitochondria at all. That happens out in the cytoplasm of the cell. Step two. Anybody know what step two of respiration is? Named after a person? No? It's called the Krebs cycle. You've all heard of that before, right? Krebs cycle? That happens in the middle part of the mitochondria, purple area. That's called the matrix. It's like the center of the mitochondria. Happens in the matrix. And then the last part has got a couple of different names. The last part is sometimes called the electron transport chain. I prefer, though, for this class, to call it something a little bit different. We're going to call it We're going to call it oxidative phosphorylation. Now, that's a big sounding word. We'll get into what that means later. But oxidative phosphorylation or electron transport happens on this inner membrane. And by the way, these folds in this inner membrane are called cristae. That's a word you need to know now. The cristae is the name for the folds on that inner membrane, which is where electron transport happens, or oxidative phosphorylation.
So they're the same thing?
Yeah, we're going to use these two words interchangeably.
Okay.
I'm usually going to call it oxidative phosphorylation because it's more descriptive. Let me back up to that previous slide. Back to something else, that whole structure determines function thing. We said that every cell basically has all the organelles, but they don't necessarily have what? The same amount. Some cells are loaded with mitochondria. Your muscle cells are loaded with mitochondria. Because they're the cells in your body that probably use what? The most energy. Little sperm cells are loaded with mitochondria. They have to do a lot of swimming. It takes a lot of energy to do that. Other cells don't have nearly as many mitochondria. Also remember that mitochondria have their own DNA. And what does that DNA look like? Looks like prokaryotic because how many chromosomes are there? Just one. And that, what shape is that one chromosome? Yep. It's a circle. For you, when you inherit mitochondrial DNA, which parents do you inherit it from? Always. You get one extra chromosome from your mom that you don't get from your dad. You get 23 nuclear chromosomes from your dad. That's what the sperm delivers. You get 23 nuclear chromosomes from your mom. That's what the egg delivers. But what else does the egg deliver that the sperm doesn't? All your cells what? Not just some chromosomes, it also delivers what else? Organelles, like the mitochondria. Organelles come from mom, not dad. All right, let's wind up new stuff for today. Probably look at this slide. Question?
I missed the last part about the oxidative phosphorylation. That happens in the cristae?
It happens on the cristae, on the membrane.
That's the actual membrane?
Yes.
And the matrix is a part inside of it?
Yes, it's that area in the picture that was purple. And here. And that's where the Krebs are.
Okay. And then the cristae is the membrane?
The actual folds around it. All right, winding up, last couple things here with plastids. We're going to say that plastids are bags of pigments. What's a pigment? Okay, that's normally what we think is a color, but I don't want you to think that. I want you to think a pigment is a protein. A protein that does what? Why are pigments different colors? Yeah, but what are they doing to make them be different colors? Why is chlorophyll green and another pigment orange? Why is her shirt green? And Joy's shirt blue? And Christina's overshirt white? Exactly. Different pigments absorb different colors of light. Christina's shirt there being white. What's it doing with light? When white light hits Christina's shirt, basically all of it what? Reflects. And when we see all colors of light in equal amounts, our brain interprets that as what? White. On the other hand, anybody got on black? Yeah, there's a black sweatshirt. When white light hits the black sweatshirt back and back, what happens? It gets absorbed. Not hardly anything gets reflected. And when our brain sees no light coming from something, our brain interprets that as what? Black. On the other hand, when white light hits the green shirt here. Yeah, just green. I was messing my colors up. When it hits the green shirt, what happens to the white light when it hits the green shirt? Really, light is really made of what 3 colors? That light coming out of those bulbs, if we boil it down to it, it's really just made of 3 colors. What are the 3 primary colors of light? All other colors can be made from. It's different than paint. Red, help me out. Not yellow. Not purple. I've got a set of spotlights I'll bring in here and show this to you later when we get to photosynthesis. Red, blue, and green. All colors of light can be made from red, blue, and green. When white light hits the Avengers t-shirt there, or X-Men, or Marvel, or whatever kind of t-shirt it is, what happens to the red and the blue? It absorbs. What happens to the green? It gets reflected. Our eyes see what gets reflected. So that shirt's reflecting green. Joy's shirt's reflecting blue. If you're wearing a black shirt, what's going to happen when you go out on a really sunny day? You're going to get hot. Because if it's getting absorbed, it's getting converted from light to what? Heat. You go out with a white shirt, you're going to feel pretty nice, right? Because what's happening to that light when it hits your shirt? It's getting reflected. So tie it back into this slide. I want you to know that pigments are proteins. They're proteins that absorb some kinds of light, reflect others. Chlorophyll's green. We know that's the main plant pigment. What would that mean if we had a room full of plants and the only light in here was green? What would happen? If we only had green light, we had a room full of plants. What would happen with the plants? Would it be good or bad for the plants? It'd be bad, because when the green light hit them, what would happen? When the green light hits the plants, what would happen? It would bounce off, which means they wouldn't be getting energy to do what? Photosynthesis. What color would the plants appear to your eyes if you walked in? So room full of plants. The bulbs are green. What color would the plants appear to you? Green. That's what happens when the green light hits the plants. Reflects. And what do you see? You see what gets reflected. Now, on the other hand, if we were in a room with only blue bulbs, plants, only blue light, good or bad for the plants? They're probably good. Turns out plants like blue light better than any other color. Helps them grow better. You walk into the room, what color are the plants? Oh, they're kind of black. Because what happens when blue hits a plant? It's absorbed. What's getting reflected? There's only blue light, and blue's getting absorbed, what's getting reflected? Not anything. What does your brain see when it's not seeing any light? Make sense? All right, so we know the chloroplast, that's an example of what we call plastid. We know that's where photosynthesis happens. Big picture. Photosynthesis uses what to make what? Uses light energy to make, so let's don't say make. Let's say it converts light energy into what kind of energy? Usable. Okay, let's do a little more exact. Usable is a good word though. We're going to say light energy to chemical energy. Chemical energy in what form? What chemical does photosynthesis make? Sugar, glucose. What other two things does the plant need to be able to do photosynthesis, other than light? What are the other two main ingredients, reactants for photosynthesis? Water and carbon what? Carbon dioxide. Water and carbon dioxide. Question?
Okay, so the plastics, the chloroplasts are in the plastics?
No. Plastids are bags of pigments. A chloroplast is a type of plastic.
Oh, okay.
It's a chlorophyll containing plastic.
Okay.
It's a type of plastic. That's a good question. All right, let's go to a picture of a chloroplast. I think I have one on here. Maybe I don't. I'll draw one. Yeah, my drawing's terrible. All right, so here is our chloroplast. Inside our chloroplast, we're going to have all these little bag-like structures. My picture doesn't show this, but like a mitochondria, the chloroplast basically has a double membrane, a double phospholipid membrane. It also has its own DNA, which looks like what? Bacterial, single circular chromosome. More proof of what? Yeah, that there is called the endo what? No. Endosymbiotic. That eukaryotic organelles started out as what? Bacteria that got engulfed by bigger bacteria. And then over time it worked out to be a good deal for both. And that gave way to eukaryotic cells. Regardless, inside our chloroplast, each one of these little sac-like structures is called a thylakoid. Those are thylakoids. That's where the chlorophyll is at. The chlorophyll is actually embedded in the membranes of these little thylakoids. Maybe those are pieces of chlorophyll. What machine that people use is chlorophyll sort of like? A bunch of these on the South Parkway. Solar panels. A lot of solar panels. They absorb light and turn that light into some kind of usable energy, right? Light to usable energy. Think of chlorophyll that way. But the thylakoid, each one of these bags is called a thylakoid. A stack of these thylakoids, the whole stack, is called a granum. What's the difference between granum and grana? It's a singular plural kind of thing, right? Granum is singular, it's one stack. Grana are multiple stacks. And then this area between the grana is called stroma. And we'll get into how all that works later. But again, make sure you know that chloroplasts contain their own DNA. They have their own ribosomes. They have a double membrane. More proof of that whole idea of endosymbiosis. They also can do what independently of the cell that they're in? What can they do when the cell that they're in is necessarily not doing it? Mitochondria can do this as well. They can split. And how do they split? How bacteria split. Bacterial reproduction called most of the time. So one bacteria splits into two bacteria. Binary fission. Chloroplasts and mitochondria basically can do binary fission. All right, I'm going to skip this cytoskeleton stuff for the time being. I'll come back to it. I want to mention this one, and then we'll be done. Peroxisomes. Peroxisomes are another membranous sac-like organelle. They have inside them enzymes that help our body to detoxify some of the products that are made during cell respiration that are dangerous to us. One of those products is hydrogen peroxide. Your body makes hydrogen peroxide, which is toxic. Your body needs to be able to get rid of it. An enzyme that we just talked about at the beginning of class today, catalase, helps us to get rid of peroxide. But what you need to know about peroxisomes is basically this. They're membranous sacs. They have enzymes that help us to get rid of toxins, mostly toxins that were made during cell respiration. They also help to rid our bodies of free radicals, which we'll get to later in terms of what those are. But I just want to make note, help to get rid of free radicals. Well, I'll explain to you a little bit later what a free radical is. All right, that's a good place to stop on new stuff. Questions? We've got a little bit more work to do on this PowerPoint in terms of finishing it up, but not too much. We're getting there. We're almost finished with that one. We turn off our recording, and I'll pull up the other Moodle assignment. We'll talk about it briefly.